All right, this is Mr. Labonte. I'm going to be talking about the process of transcription and translation. And this is the mechanism of making proteins from DNA. Obligatory cartoon. This is topoisomerase, which is a particular enzyme, and the job of it is to help unwind DNA, hence let yourself unwind. Helicase is the actual protein that splits DNA, so it looks like a little wedge that I'm drawing in there. All right, so here we go. So there's this description of this thing called the central tenet. And the central tenet is you start with DNA, DNA is translated into RNA, in this case messenger RNA, and then that is translated into a protein. So this is how proteins are formed, from DNA to mRNA, and then translated by the ribosomes into a protein. So this is what we're talking about. We're going to be looking carefully at each one of these parts. I went over this in the DNA lecture, but in case you didn't have an opportunity to look at that, we're going to start here with this idea that DNA is wound around histones. So this is a histone. This is it's a complex of really 9 different molecules, but they're all put together, and what they can do is they can hold about 147 base pairs. So DNA is wrapped around it twice and then is wrapped around another histone. And this allows DNA to be condensed into the nucleus, which is really fairly small, 25 microns for something that's 3 times 10 to the 9th base pairs long. There's a lot of winding up that happens. This is important because DNA cannot be transcribed or replicated, for that matter, unless it is unwound from the histones. So there are some areas of DNA that are permanently unwound from histones, and these are enhanced regions. And there are some areas of DNA that are very easily unwound from histones, and these are hypersensitive regions. So as you may imagine, these are going to code for proteins that you need them every day. This is another more focused look at histones. These are the little tails. That was that yellow part. And you can actually acetylate these histones, and this is a mechanism actually of locking them, and you would never be able to make proteins out of the DNA if those are locked down. So this is just another example of that, or methylated is another way of calling that. And so these are these methyl groups that go and can get locked onto histones. And this can happen. You can actually inherit these things, and they can cause genetic issues for you. Okay, so this question is very important. So I can't express how important this is. Because your DNA codes for every single part of your body, and you have your full complement of DNA in every single one of your cells, the vast majority of your DNA is never used in any given cell. So for example, you don't want to be making an eyeball in your ear, right? But you have the DNA to make eyeballs, and it is in your ear. So you need to have very specific promoters that are going to tell specific parts of the DNA to open up and to code for particular things. And in general, the way of thinking about this is that DNA is locked down. So nothing is going to happen in DNA unless it's very specifically promoted. This was first discovered in bacteria. So this is an example of bacteria. You have this what's called a regulatory gene, and this gene is made into mRNA, which is made into this protein. The protein bonds onto DNA and locks that DNA down. Now it's only when you have something called an inducer, so this would be something from the environment, that comes in, that inducer is going to bond, remove that lock, and then you're going to be able to code the DNA. So basically like riding a bike. You can't ride a bike unless you unlock it first. So that's the thought I want you to have. In this goes over all of the different steps. Again, this is kind of the central tenet. So you go from DNA to RNA. The RNA is copied, it's processed, it moves into the cytoplasm. So I'll give you different numbers. I'll put the little numbers next to each of these. This number 1 corresponds to this number 1. That's the copying. This number 2 corresponds to that 2. That's the processing. This is 3 moving out. This would be 4. This is when amino acid, oh, sorry, excuse me, that is not 4. Let's erase that. Here's 4. These are amino acids being attached to the tRNA. And then this is 5 tRNA strands being attached to make this. This thing that I just highlighted here, this is the goal, right? This is a protein. So that's the string of amino acids. That's what we're going for. So this is an overview of everything. In this lecture, actually, we're only going to be talking about this first part, everything that's going on in the nucleus. So going back to activators, in eukaryotic cells, you can see the activator occurs what's called upstream of the DNA. This is UAS, upstream activator system. There's this spot right here, which is called a promoter region, and it's got this great thing, TATA box. And you'll remember, I think we talked about there's only 2 bonds between thymine and adenine. And so you're going to have fewer hydrogen bonds here. So this is an ideal place to start splitting the DNA. The TATA box is going to have all these different proteins that are going to bond on. But this protein in particular is quite important. And this protein is RNA polymerase, PO. So what does it do? It makes RNA. So it's a protein that makes RNA. When you have all of these specific promoters that come together in a specific place, that's what's going to allow RNA polymerase to bond. Now, what are these promoters? These are all signals that the cell is receiving that this very specific protein is needed. And so the cell is not going to make the protein unless all the correct signals get there. So this gives the cell a lot of flexibility in terms of what it makes, and also the quantity of what it makes. So it allows it to dial it in and out. Once the RNA polymerase is attached, it's going to move quite rapidly along the DNA, and you'll notice here, I hope, I'll show you this, it's only on one strand of the DNA, so this is called the coding strand, to make the copy of mRNA. This is a close-up region and a little more simplified image. This gray ball here, this is the RNA polymerase. Okay, so this is a large protein. It's going to be moving from left to right, and it's going to be on what's called the template strand of DNA. So DNA is getting what's called unzipped. Here, remember that helicase from the cartoon, so that's helping relax the DNA and unzip the DNA. The RNA polymerase is moving from here, so this is on this side, from the 3' side to the 5' side of this particular strand of DNA. So it always moves from 3' to 5', and as it does, it matches nucleotides. Adenine is coded for thymine, and remember, there is no thymine in RNA, so uracil is the complement to adenine. And so this strand of nucleotides is generated and grows goes longer and longer. And then as the RNA polymerase passes, the two sides of the DNA, the nucleotides of DNA, anneal back together or melt back together and close up. This is just showing you, again, you're moving from 3' to 5', and because it's anti-parallel, what you're creating in the RNA is a 5' to 3' side, moving again from left to right. This just is talking about starting at the ta-ta box and the fact that you have, sorry, not 3, but 2 hydrogen bonds per nucleotide. So it's going to start there. And then this shows you, this talks about the speed at which this happens, and then there are a couple videos that I'll show you in class that really give you a sense of this. So this is 1,000 nucleotides a minute, or 16 nucleotides per second. So this is happening very, very quickly. But if you think that, right, I think I said to you that a protein is on average 240 amino acids long. If you're making these proteins, you do need to move, the cells need to do this rather rapidly. All right, once the entire strand is made, and what happens is the RNA polymerase will get to the end, there'll be what's called a stop codon. It will sense that there's no more DNA. I mean, there's still a ton more DNA, but there are basically these weak bonds. It'll cause RNA polymerase to break off. The mRNA will go out, and then you'll have a long strand of mRNA. So here's the mRNA. And this strand of the mRNA then actually gets edited. And what happens is you have these intervening segments called introns, and they get kicked out and cut. And so you only have the exons, exogenous components that are good and useful. So it's often asked why you would have these intervening segments. So to kind of understand this, let me give this DNA, each of these exons and introns, a letter. So we're going to do L-E-A-R-N. And so all together, if we didn't take any of these segments out, we would have the word learn. And so maybe this would be what was translated, and that's possible sometimes. But you could see that you could take the R and the N out, so these could in fact be your introns. You could take those two out, and then you would have Leah, right? Alternately, if you did not want to take those out, you could keep those in. And you could take the L out, you could take that out, the N out, and you would have ear, right? But you could have those back in, and you could take the R and the E out, and you could have LAN. So by having these introns and exons and being able to pull different ones out, you get a lot more flexibility in the protein. So that's one reason. So I'll circle that and I'll write 1, flexibility, flex, so you get flexibility. But the second answer is that some of these are always taken out. So we'll do this one in red. And the reason that some of these are always taken out is they are actually put in there by viruses to code for viral proteins. And so the body has learned to basically to cut these strands of DNA out and never code them when they want them. So that's the second reason. So finally, after you have your introns and exons taken out, the last thing that's going to happen is you're going to put a 5' cap. So there's a guanine that gets flipped around. So basically you have 3' on one side and another 3'. And you have something called a poly A tail, which is put on the other end. The reason that this occurs is once the mRNA goes out into the cytoplasm, it is going to be subject to being destroyed by enzymes that are in the cell, and the job of those enzymes is to destroy mRNA. Because viruses can use mRNA, as we're going to learn a lot when we talk about COVID. And so our mRNA, which is good mRNA and has a specific job, needs to be protected from enzymes that destroy mRNA. And this 5' cap helps protect it, and the poly A tail helps protect it as well. And if we have time in class, I'll talk a little bit more about how that works. All right, I hope that helps. So this finishes transcription. So you have, first of all, a need in the cell, or the presence of something that's going to give you a promoter. That promoter, which we're just going to call P now, is going to help attach RNA polymerase, which is going to do the translation. So then we're going to do translation. And then finally, we are going to process. So this is the fourth step. We're going to process the mRNA. And what does process mean? It means we're going to take out the introns and exons, and we're going to do the 5' cap poly A tail. There it is.